Hello everyone, in this video we are going to be taking a closer look at the gene expression omnibus library, and specifically using the GEO2R data analysis tool to observe trends in our data. Recall that in our first video we had established criteria for our dataset, and we actually found a dataset that worked and fit our needs. Some of the major things that we will learn in this video include number 1, data analysis, number 2, data manipulation, 3, statistical significance in gene expression data, and finally 4, we will hone down on key genes and establish our final dataset that we can use for our project. So without further ado, let's hop back right into our gene dataset. Okay, so here we are in our gene dataset. Now, if this screen does not look familiar, or if you're not familiar with Geo datasets, please go back to the previous video in this channel and skip towards the end where we actually navigate to the screen and find our dataset. Now, as you remember, this is our dataset, the one with the 29 samples, and what you want to do is click this GSE number. So instead of normally, right, if you see a page like this, you would most likely click this title. But once you click this title, it takes you to the overall summary page, where it gives you a summary of the actual study that was done, and it gives you the citations. But what we want is the actual gene dataset itself. So we actually click the series number, which in this case is GSE 21942. Now, once we click on that and we scroll down to the bottom, we can see this large button right here, which says Analyze with GEO2R. Now, what exactly is GEO2R? GEO2R is a publicly available tool that is used to analyze genetic information. Specifically, it allows researchers, like ourselves, to compare 2 or more groups of data to see which genes are differentially expressed. Now, I can understand this might be a little bit confusing, and the terminology here can get a little advanced, so let's do a quick recap of where we are. Now, in our previous videos, specifically the one talking about the biology behind multiple sclerosis, we learned how genetic factors may play a crucial role in the formation of MS. So, tying back to our main objective, if we can see which genes are either overexpressed or underexpressed in patients, we can use this gene's level of expression as a data point for our neural network. So we can sort of visualize this here, right? Let's assume that white is healthy and this orange sort of color is diseased. Soon, the neural network can sort of see, gene A in a normal patient seems to be very lowly, its level of expression is quite low, but in a patient diagnosed with MS, that same gene A, is very upregulated. Now, gene B, on the other hand, another data point in the neural network, could say, in a normal patient, it seems to be very upregulated, right? The level of expression in a normal patient seems to be very high, gene B. But in a patient with MS, this same gene seems to be downregulated quite a bit. Now, once the neural network then sees this sort of pattern numerous times, it can identify this as something very common in people with multiple sclerosis. So let's see how we can incorporate this data into a neural network. So first, let's go back to that GEO2R tool. All right, and we are back in this GEO2R dataset. Now, a key thing to point out, this course is designed for you to, it's designed to allow you to use what we teach you here for all types of neurological diseases with a certain genetic factor. However, if you choose to follow our method, you need to be sure that there's an Analyze with GEO2R button here. If it's not present in your dataset, then you will have to choose another dataset or figure out a different way to make a neural network, because in our methodology, we require the use of the Analyze with GEO2R tool because it is a very, very powerful tool that allows for the comparison of different gene datasets very effectively. And there might be some different softwares out there that are able to do this as well, but I'm only aware of the Analyze with GEO2R one, and it is a very simple one since it connects right into the Geo expression omnibus, and it's very simple. So with that, let us now go ahead. So click the Analyze with GEO2R tool, and it will lead you to a screen where you see all of the data sets already here. And actually, I got ahead and already went started. So let me just close this. Okay. So this is what you would be seeing. You would be seeing a lot of samples. In our case, we have 29, but usually whatever number of samples you have in that data set which you selected would be seen here. So now obviously the first thing that we need to do if we are to compare the two datasets is to establish our controls and our disease datasets. Now recall that in a scientific experiment, our controls are basically our base value. That is the healthy patient here. Now how we do this is we go to define groups and we type in control. Now what is vitally important here is that we need to establish our control first. Always our control first. This Analyze with GEO2R tool works in the way that it identifies your control sample as the very first group that you identify. So whenever you do this on your own, make sure that your controls are always the first group that you identify. Okay, so now typically in a normal data set, right, you would have a different title, perhaps. Ours is very organized in the sense that it has control 1, control 2, control 3, and so on. But usually the titles may be a little different. It might be a little bit scattered. The controls might not be all lined up. So you can just sort it with the title name. And then it automatically puts all the controls in alphabetical order. But yeah, it just makes the whole process a little bit easier. Okay, so now that we have our control group defined, let us actually input all of the control data sets into that. So what you want to do is click down on the control one, click shift on your keyboard, and go all the way to your last control. In our case, this is control 9. But yeah, we should just have all the controls in this yellow format. Then go back to the define groups and click on control. Now what we should see is all of your things labeled control should be in this green sort of color, and there should be a label and a group association which says control. So now what we've done is we've taken all of our controls in our data set and we've established them as controls. Pretty simple. But now comes the second part where we need to do the same thing for our diseased, right? Because we have a control and we want to compare that to the diseased data set. So take this time to pause the video and try to see if you can do this on your own. It's the same sort of methodology that we used for the controls, just do the same thing as a mini quiz for you for the diseased one. Okay, so I'm just going to assume that you did it, but I'll walk you through it just in case. So here we have, right, the first thing that we need to do is define the new group. Let's call it MS patients. So that is our second control, not second control, but it is the disease group. Click MS patient 1, click shift, go all the way down to the bottom, and put it in the MS patient group. Now what we have is success. We have a green control and a sort of purplish violet or sort of hue. I'm not too sure what this color is. I'll just call it purple for now, but it is a purple MS patient. So what we've done is we have effectively and efficiently put all of our controls in the control group, all of the MS patients in the MS patients group. Now what we've done is we've established our data sets, and as we scroll down, we can actually click this button here, which is analyze. And what this button does is it analyzes the differences in gene expression values from the control versus the MS patients. Now, usually this takes a while, so as we wait for the magic to happen, I want to point out that this data that we get in this project is hyper-specific to MS. The genes we talk about here may not be important in other diseases. So if you are interested in using this method for another disease, do not just use whichever genes we find as significant, but use this entire method, because it's very important to see which genes are important for your specific project. Okay. So now what we have is a large list of table values that at first can look a little bit intimidating, so let's walk through the important pieces one at a time. Firstly, log FC. Now, log FC stands for a mathematical principle known as log fold change, which means that it is the log base to fold change. Now, what we can do is take 2 and raise it to the log fold change value. Our answer is actually the ratio of expression. So that's a little complicated. So for example, this HINT gene here, 2 raised to 3.101 gives us around 8.58. Now we can say that the gene HINT is on average 8.58 times more expressed in a normal person compared to an MS patient. Taking this into consideration, if the log FC value is positive, that indicates that it is overexpressed in the control, whereas a negative log FC value indicates that it is underexpressed in the control. Next, our p-value indicates to us how likely our data is based on chance and random luck. So, taking a step back, there is something in science called the null hypothesis. The null hypothesis observes no real change. For example, in our case, the null hypothesis would indicate that there is no difference in expression in gene values between a control and MS patient. That's our sort of baseline value. Our p-value is the strength of our argument in order to reject the null hypothesis. Think of this as a court trial. In order to find someone guilty, there's a certain threshold of evidence that is needed to be provided. Fail to meet that threshold, and you will not win the case. In that same way, the p-value either fails to reject the null hypothesis, meaning that there is no difference in gene expression, or it does reject it, meaning that there is. And the lower our p-value, the stronger our court case gets. Now, I understand that this is a fairly complex topic, so I highly suggest you take a deeper look at things such as the chi-square test, as these are crucial topics for scientific support, especially when it comes to machine learning. Anyway, we want our p-values to be low. A p-value lower than 0.05 is considered to be the threshold. Anything lower than that is excellent. And in our data, we can sort of see that we are a-okay in our regard as our p-values seem to be way less than 0.05. So we're okay with that. Now what we want to do is look at all our data more in depth. So to do that, go up and download the full table and upload it into a Google Sheet. I'll see you guys soon. Alright, so once you've gone and uploaded your dataset to a Google Sheet like this, you can sort of get a grasp on how large our dataset actually is. If we scroll all the way to the bottom, you can see we actually have over 54,676 genes, which is quite a lot for any neural network, but we'll get into that a little bit later. And actually, if we scroll all the way to the bottom, we can sort of see that our p-value is incredibly high, and our log fold change value is incredibly low. So we can sort of tell that these genes at the bottom here are really sort of irrelevant to our dataset and don't really contribute much. So we need some sort of way to refine our dataset to see which genes are actually important for our dataset. So to sort all this out, first let's go to our top row, go to View, Freeze, and freeze the row. So this row is now frozen, so we can go and play around with our data a little bit more. So the first way to hone down our data is p-value. As we said before, we want our p-values to be less than 0.05. What we can do is we can scroll down to the place where our p-value is less than 0.05. So if we keep scrolling, let me just scroll like this. So we should be getting quite close. So this is 0.6. All right, here, somewhere around here. There you go. Okay, right here. So we can see right here, it sort of transitions over, right? So what we can do is anything less than this, we can actually just delete. So if we just click Control Shift and then scroll all the way to our bottom of our data set, Shift, just simply delete. This might take a second, but once it's done, you can see we have reduced our data set to a mere fraction of the size it was earlier simply by looking at our p-value. Now that we've done that, we can now take a look at log fold change value and see how our data compares with that. So let's take a look at that now. Alright, so taking now a closer look at log FC value, right? We had briefly discussed how log FC value is, long story short, it's just a comparison between the gene level of expression in a healthy patient versus a patient with MS, right? So this is really the key data point in our neural network. So we want to see the vast differences in expression levels. So what we can do is we can actually click on this column, go down here and sort from A through Z. So these are all super negative log FC values. And what we want to do is we want to take maybe the top 15 ones that are most significant. So what we can do is we can actually just Command Shift Control C all these values, create a new sheet, and just dump it all here. So now what we've done is we've identified the top genes that are very different with negative log FC values. Now we can do the same thing with our positives. So go from Z to A. And here you can sort of see we have a lot of empty data set. And sometimes the gene analysis people in the GEO2R dataset sort of do this to fill in some of the missing gaps, but we can just delete some of that. So just to make our data a little bit cleaner, the last one, I think. Okay, cool. So now we've sort of taken a closer look at the positive values, and we can sort of select the first top 15 as well. So if we just do the same thing, select, Control C, we go to our double sheet, we can click Control V. And now what we have is we have the top positive log FC values and we have the top negative FC values. And these are really going to be our data sets for, and these are the important genes that we're going to be analyzing in our neural network. And it's very important to see that, one of our main objectives for this project was to identify key genes that play an important role in multiple sclerosis. And quite frankly, we've already sort of done that. We've identified around 28 genes here that play a statistically significant role in MS purely based on their log FC values. Given the fact that they are highly overexpressed or underexpressed, given what log FC value you look at, right? We've identified that these specific genes play a crucial role in MS. But now we want to get into the prediction of it, right? Our fundamental objective in this project is to be able to predict early onset of multiple sclerosis and not just identify a few key genes. So how exactly do we do this? Let's get into it. Okay, so let's just sort of take a step back and recap exactly what we've done and what we want to do. So what we've done is we've taken a look at the general data set that was given to us by the GEO2R database. Now what we've done is we've simplified it down based on our p-values, right, to just get it down to a fraction of the size it was before. And we've reduced that simplified data set even more by looking at the log FC values to identify some of the genes that we think are statistically significant in multiple sclerosis. So if we just copy paste these genes, right, and we go to a new data set, these are the genes that we have identified as important. That's pretty much it. In the simplest terms possible, we think that these genes are important. But now, what exactly do we do with these genes? Let's take a look. What we want to do is we want to find the values of expression for each of the genes that we had identified and compare them with the other values in the data set. So what that means is if we go back to our data set right here, right, in Gene Expression Omnibus, we have 29 samples in our data set. 15 of them are control and about 14 of them are MS. So if we actually click on the control one here, right, and we go down and say view full table, this sort of looks intimidating, but don't worry, I'll walk you through it. What these are, what the ID/REF values are just a fancy way of identifying a gene. So if we go back to our data set, right, at number 2, let's say we want to find the value of expression in the control 1 data set for the HBD gene. We go and click the ID/REF value, go to our data set, Command F, and search for that. Now we know that in control 1, the HBD gene has a value of expression of 123.9. That is one data point in our neural network. So if we go back, I've basically taken all of our genes that we've identified as important and I've transposed them and I've put them all into rows. And what I have at the last row is the outcome. This outcome, of course, refers to if the patient has MS or not. So if it is MS, we can say 1, and if it's healthy, which we're looking at the first control right now, so that means it is healthy, we can say 0. And what we can do is we've identified the HBD gene to be 123.9, so we can just go back to our data set, copy paste that. Now let's try LTF. We go back to our ID/REF value. LTF has an ID/REF of this, copy paste that. Let's search for that now. It's 12.9, so we can copy paste that. Go to our data set, TF12.9. Now, when there is something like this, all you need to do is just delete this first one, and you have this. So what that means is, I'll just do one last one with you. If we just do this, HBD. So what we can do, sorry, wrong data set. If we go back here, select this, go back here, search for it. We have 13.9, copy paste that. Go to our data set, paste that in. Now take a minute, pause the video, and see if you can do this one, this LAAS2 gene, and try to find the data point, the data value of expression for the control 1 dataset all on your own. So pause the video now. Okay, cool. So let's take a look at it. So ALAS2, we can go back here, select the ID REF value, go to our control 1, search for it, select the value of expression, go back to our transpose dataset, enter that, and we've done that. So now, as we keep on doing that for each of the genes, right, we'll eventually have one row in our column. And if we keep doing that for all 15 of our controls, and then all 14 of our MS, we will eventually have a healthy amount of data that we can use for our neural network. Now, I'm not going to I'm expecting you to do this all on your own, but in case you find it a little bit difficult, I'll have the end result linked to this video. And I'm just going to go to that right now. And it's just this, and basically all I did was I just did the same thing that I've just walked you through. I went and found the ID REF value, gone and compared that to the control value dataset, and I've entered it into our dataset. So let's take a look at what the final values will be. All right, so here we are. I might have edited the data a little bit just to make it a little bit easier for you guys to understand, and I might have changed a few genes, but this is basically it. We have our final data set available to us. We have each of the genes, each of their values of expression, and the correlating outcome of that expression value. So what we can do is we can take this gene data set and we can actually use it as data for our neural network. So super exciting stuff. And what we've done, just to recap, we've taken all these values, we've seen their levels of expression in each of the controls and each of the MS values, and we've placed them here, and we've labeled the outcome as either healthy or diseased. Healthy indicating a 0, 1 indicating a diseased patient. So this was super exciting, and let's go to a general recap right now. All right, guys, let's go over a quick summary of what we've done. So far we have provided a large amount of data manipulation, right? We've looked at log FC values, P values, etc., which are major terms in science, and not just for, machine learning, but these are comparison tools that are used in all types of science, in all different fields. So it's very important that you have a good grasp of them if you want to continue this project outside. Next, some of the things that we've completed for this project in specific, we have secured our data, we have shortened our data using these log FC values, we have observed specific genes that may play a crucial role in MS, and finally we have our final data set acquired for our project. Now, I will be linking the final data set that I am going to be using below, but you can just sort of follow the method that I had discussed to create your own data set. But I highly recommend using my data set just so you can follow along and you don't have any extraneous data. Now, upcoming for the rest of our project is finally we're going to be getting into the programming. We're going to be discussing Python, we're going to be discussing machine learning, logistic regression, all sorts of really fun stuff that is really at the forefront of technology in today's world. So I'm really excited to get started with that with you guys. So be tuned and see you soon. Thanks.